All right, chapter 10 is about the structure and function of DNA. This will be our last chapter with cellular stuff, so we'll move on to other things. I don't know about you all, but I'm excited that we are going on past this. All right, so DNA structure. If you look at the chapter in the book, it's a lot of material, but we're just going to use just a little bit, and then we're going on to chapter 13. So DNA is made up of a sugar called deoxyribose, and then a phosphate group, and then there's a nitrogenous base, a base that has a nitrogen as its foundation. And each nucleotide, which is all of this, the sugar and the phosphate group and the base together, it's called a nucleotide. Each nucleotide is made up of a different, or one of four bases, right? One of four bases. There's adenine, we'll use A, thymine, which is T, guanine, which is G, and cytosine, which is C. Okay? And so this DNA structure was first discovered not until 1953 by Watson and Crick. They won the Nobel Prize in 1962, 56 years ago. So it hasn't been that long ago before we even knew, we didn't even know what DNA looked like, right? It's held together by hydrogen bonds that keep the two strands, because it's a double strand, together, these little hydrogen bonds. And these hydrogen bonds form between these nitrogenous bases that we talked about, adenine, thymine, cytosine, and guanine. Okay? If on one strand of DNA there is an A, adenine, then on the opposite strand, opposite it, is always going to be thymine. Likewise, if it's thymine, on the other strand, it's always going to be adenine. So A and T match up together. Likewise, cytosine on one strand, then guanine's going to be opposite it on the other strand. If it's guanine, then it's going to be cytosine on the opposite strand. So they always pair up this way. This pairing is constant among all species, but how they are put together on the DNA strand is how that gives each species and each individual of each species their individuality, right? So if we draw this real quick. So it's DNA is a double-stranded helical, like a winding staircase, all right? It's hard to draw on 2 dimensions, but if we have bases over here, right? On the opposite strand then, you'll have their complements. And they're held together by these hydrogen bonds, right? Okay? Now, we talked about DNA replication. Remember that? I forgot. So, on your exam, what I'm going to give you is something like this, where I'll give you the DNA bases of one strand, okay? And what I'll do is I'll ask you to match those bases up, either above or below. That would be on the other strand. So in this case, remember that C and G always match each other. So right here we have a G and C again. So a G here. Now we've got a G, so on this strand there would be a C. A with T, T with A. C, G, G, T, T, C, A, G, T. Right? So that's all you have to do. I will give you one strand, and I want you to put the bases together below or above, it doesn't matter if it's above or below, to match those bases on the other strand. Okay? So this next slide just shows those bases underneath, just like that. And that's what you will be asked to do, all right, on your next exam. All right. So remember DNA replication. That's where during the S subphase we have the DNA molecules making a copy of themselves. So if we go back to this strand over here, all right? So with the help of enzymes, this double helix unzips, all right? So now you've got like this, all right? So on one strand, we have T, G, G, and if we're looking at this one, and it comes up here, it's T, A, G, C, right? And then this strand starts here, goes this way, is A, C, and C, and then over here it's A, T, C, and G, all right? So they're unzipped now, all right? And what happens is that the nucleus always has a supply of these bases, like a construction site. You always have all the material stored up so that you don't have to run all over the place to find them. So, and that's what happens with these bases, okay? And so what happens is it starts, let me move this just a little bit further apart. All right? And so what it does during the S subphase is it just starts matching bases. It has nucleotides right at its where it can get to them. There we go, John. So it just starts matching them up. Once it starts matching them up, these start twisting again, right? And so as it unzips, it zips right back up. And when you have that, what's left is the actual exact copies of the ones you started out. So that's how replication works. All right, you won't have to do anything with that on your test. I just thought that might be something that you would sort of need how it works. The last part of the chapter in the book deals with protein synthesis or production, and this is a long process. There's many things going on, and it's just too much. I want to move on, and so I boiled it down to two words, right? Transcription and translation, right? Transcription is where enzymes assemble the RNA, all right? So now RNA is involved. onto a DNA template, and this template then is used as a blueprint to build a particular protein. All right. So let's say that on our strand here, ACC is the part of the DNA that needs to be coded in transcription onto RNA so that we can get the right protein made. All right, and so what will happen is the DNA will unzip wherever it needs to. We have ACC here, all right? And that's what we need. And so it will take RNA nucleotides and match them up. So G and C, they use the same nucleotides. Now, instead of thymine on RNA, they don't use thymine. They use another one called uracil. But don't really, I don't want to get you frustrated on the part we talked about where you write down your the bases under the other, one strand of DNA to the other. So we'll just go ahead and use T, even though it really is a different one called uracil, but we're not going to use the. All right. So now the information that tells us what protein is going to be made is now the opposite of what's on DNA, right? But that information is transcribed onto the RNA molecule. Now the RNA molecule, there are several of these that interact with each other. And finally, the RNA is taken to ribosome, and there's ribosome RNA there. And here's our T, G, and G, right? And what happens is that the ribosome has RNA bases at its disposal, just like the nucleus, and it then puts A, C, and C together, and when it does, it translates the transcribed information, and now we know what protein is going to be made, ACC, all right? And this is called translation. So transcription is like if you're going to build a house and your idea of what you want your house to look like is in your brain, right? And you go to an architect and you tell the architect, this is my idea of the house. The architect takes the information from your brain, your idea, and puts it onto a blueprint, right? So he transcribes your idea onto paper. When you want to build the house, you take that blueprint and you go to the builder or the contractor and you hand that blueprint to him, and then he translates what's on the paper into a house. All right, he builds your house. So this is how sort of like transcription and translation work. You have DNA. On the DNA, there's information about the protein to be made. So that information is transcribed onto RNA. There are several RNAs that interact with each other that translate that transcribed information from the DNA and make the protein. All right. So transcription and translation. And that, my friends, is chapter 10. All right.